Infections are the scourge of hospitals and other medical facilities everywhere, and the bacterial infection MRSA is one of the nastiest infections there is. It is antibiotic resistant and it can be deadly. But some USF researchers say there may be hope coming from what at first might seem an unlikely source. Hedel Gandhi has our story.
Although doctors have been using antibiotics to treat bacterial infections since the 1940s, bacterial disease remains one of the leading causes of death worldwide. Methicillin-resistant Staphylococcus aureus, or MRSA, is one of the most dangerous bacterial infections because most of the drugs used to treat it quickly become ineffective.
Methicillin-resistant Staph aureus, or MRSA, is probably the most well-known of the antibiotic-resistant bacteria, developing resistance to almost every new antibiotic in the clinic within about 12 months, resistant to almost everything that we have available to us, and kills tens of thousands of people every year in this country. And so a serious problem, killing an awful lot of people with very few treatment options.
Methicillin has sort of been the main drug for treatment of Staphylococcus aureus, and with increasing resistance towards antibiotics, it has now become, Staphylococcus aureus has now become resistant to methicillin. And so there's very few treatment options anymore for these MRSA infections. There's only one drug that is currently still effective, and it's vancomycin, and it still has a number of strains that are resistant to it. MRSA is a very important public health concern in terms of coming up with new treatments for it.
What makes MRSA especially dangerous is its ability to attack all parts of the body.
So most bacteria will cause one or two kinds of infections and do them well. This is a bacteria that can cause infection everywhere and do it well. And so it's almost unique amongst the bacteria in that it can cause infection in the brain, heart, bones, joints, lungs, liver, anywhere in the body really.
Years ago, MRSA was found primarily in hospitals and nursing homes. It's still a problem for those areas, but today, MRSA can also be found in gyms, locker rooms, and schools.
So MRSA is present throughout the environment. You probably have some on your skin, and this is part of the problem, is once you breach the skin, you get a cut or something, you can introduce those bacteria into your bloodstream, and it can go out and infect you.
Baker and Shaw have been working together for about 6 years, looking for a treatment that could overcome MRSA's ability to mutate and develop resistance. Shaw in the laboratory and Baker as director of USF Center for Drug Discovery and Innovation.
These sponges and corals and tunicates and things, as you might imagine, living in the sea, they're surrounded by bacteria all the time. The seawater is an intense bacterial environment. And so sponges and corals and the like have their own natural defenses. These defenses are different than humans' defenses. So humans have an immune response, right? So we have antibodies and things that attack bacterial infections. Animals like sponges and corals don't have immune systems, but instead they make antibiotics. They make drugs for their own defense. And so by going out into these environments and bringing those organisms back into the lab, we can isolate their natural treatments for infections, and we can use those for our own use.
Specimens are brought back to Shaw's lab where extracts are made and tested against MRSA. According to an article Baker and Shaw co-authored in the Journal of the American Chemical Society, an extract taken from a sponge shows amazing promise, killing more than 98% of the MRSA cells. But there is still much work to be done.
It has structural features that make it look like it's not going to be very bioavailable. It's not going to distribute in a human very well. It's probably not going to be absorbed by the human very well. So one of the things that we have to do is to modify it in the laboratory. So we do chemical synthesis, not only to make the whole molecule, because in fact we can't go back to Antarctica every week when we need more sponge, right? It's pretty far away. So in fact, we want to do a synthesis of the molecule to have more material to study. But in the process of developing that synthesis, we will develop analogs of that molecule and try to make it more drug-like. So realistically, at this stage of our research project, we could expect perhaps another 10 to 15 years before we will actually have a molecule that is suitable as a drug and has been through all the toxicity testing and you know, the types of experiments that one has to do to make sure that a new molecule is safe.
It's going to take a lot of hard work and effort by a big team and a lot of money, but it's something we're committed to and excited about.
For University Beat, I'm Hedel Gandhi.